Good day, Miss. My name is Marley C. Pino, BSN 1. I chose Chapter 4, which is the Bacterial Growth Requirements. Learning objectives: define microbial growth, discuss the various nutritional and physical requirements of bacteria for growth, and illustrate the bacteria growth curve with explanation of the events occurring in each phase of the bacterial growth curve. So growth. Growth, as defined in medical dictionaries, involves an orderly and organized increase in the sum of all components of the organism. The process entails the replication of all cellular structures, organelles, and components. But when we are talking about microbial growth, this is concerned with increase in the number of cells and not an increase in the size of the organism. What are the nutritional requirements of bacteria in order for it to grow? This would include carbon, nitrogen, sulfur, phosphorus, inorganic ions, and growth factors. When it comes to carbon makes up the structural backbone of skeleton of all organic molecules, and based on their carbon source, microorganisms may be classified either as autotroph or lithotroph. The second classification would be heterotroph or organotroph. Autotrophic bacteria are those that inorganic compounds or inorganic salts in order to synthesize their food. Their source of energy may be derived from light, and so they could be called photolithotroph, or their source of energy may be derived from oxidation of inorganic substances, and as such, they are referred to as chemolithotroph. Heterotrophic bacteria use organic compound like sugars or glucose. As their carbon source, their source of energy may be derived from light, and as such, they are called photoorganic drop. But if their energy source is through oxidation of inorganic substances, then we call them chemoorganotroph. Most of the important bacteria are actually chemoorganotroph. Then next is the nitrogen, sulfur, phosphorus. Nitrogen, sulfur, phosphorus are very essential for a bacteria to grow. These are essential elements needed for the synthesis of proteins and nucleic acid. For example, protein synthesis is not possible if nitrogen and sulfur is absent. Meanwhile, the nitrogen and phosphorus is needed for nucleic acid, and adenosine triphosphate synthesis or synthesize. It is for this reason that when you measure the dry weight of the bacterial cell, you'll see that 14% is made up of nitrogen, while 4% is actually composed of sulfur. Then inorganic ions needed for bacterial growth include magnesium, calcium, potassium, iron, and trace elements. Magnesium are essential to stabilize ribosomes, cell membranes, and nucleic acid. It also serves as cofactor for enzymatic activity. Calcium. Calcium, on the other hand, are needed for the synthesis of the cell walls of gram-positive bacterial. It's also a component required for the production of endospores. Potassium. Potassium is needed for the ribosome's normal functioning and the integrity, and is also involved in enzymatic reactions. And meanwhile, iron is found in the cytochrome of bacteria, which is actually part of the electron transport system or the electron transfer chain. Iron also serves as the cofactor for some enzymatic activity. Of course, let us not forget the trace elements. These are components of enzyme function of cofactors. The next is the growth factors. Growth factors are essential to promote the growth and development of the bacterial cell. These include the vitamin B complex and acids. So physical requirements include moisture or water, oxygen, temperature, and pH and osmotic conditions. So water. Um, just as this is a great deal of diversing in the metabolic properties of bacteria, there is also a great deal of diversity. Excuse me. In the types of environments in which different species of bacteria can survive, microbes are affected in different ways by their physical environment. All living organisms ways by their um, eh, all living organisms, including bacteria, require water to survive, and however, material species vary in their ability to survive in a dry environment. Although some species will die very quickly under dry conditions, others can persist for varying amounts of the time. For example, the bacterial and the spore is not affected by dry conditions and can germinate to form vegetative cells when it finds itself in a moist environment. Other non-spore formers can also persist in the environment for varying amounts of time based on their cell properties and amount of glycocalyx that they produce, but generally bacteria really require water. Then after all, bacteria cell is composed of water. And furthermore, water is the medium from which nutrients are acquired, and it's also the medium um, where metabolic activities like enzymatic activities happen. The next is oxygen. Oxygen is an important element because it is needed for cellular respiration. Cellular respiration is the process by which cells produce adenosine triphosphate, which is the energy molecule of the cell. Microbes can be classified based on the oxygen requirements, those that utilize molecular oxygen for energy production are called aerobes, while those that do not require oxygen are called anaerobes. Then some microorganisms are considered strict aerobes, meaning they strictly require oxygen for growth, and in the absence of which it will die. Obligate and aerobes, on the other hand, are microbes that cannot survive in the presence of oxygen. They do not have the enzymes that break down free radicals that are produced in the body. Then facultative organisms, on the other hand, survive both in aerobic and anaerobic conditions. Now, most medically important bacteria are facultative organisms. They are also what call as micro microaerophils. These are organisms that can grow at low oxygen environments but with diminished rates. Another group would be the capnophilus that require the addition of carbon dioxide to enhance their growth. Then the next is the temperature. Excuse me. Prokaryotes are found in all types of environment, then some microbes are able to adapt in very cold temperatures, whereas others can survive only in very hot temperature. Each species has a minimum temperature or the lowest temperature that it can grow. They have maximum temperature, which is the highest temperature for growth, and an optimal temperature, which is the temperature at which it grows. Best the ranges of temperatures that different types of microbes grow at are as follows. Then bacteria that can thrive temperature in greater than 40 degrees Celsius are called thermophilus. Those that can thrive between 20 to 40 degrees Celsius are the mesophilus. And this is where the most medically important bacteria belong. Meanwhile, the cyclophilus are those that can survive in 10 to 20 degrees centigrade or even less. Then the next one is the pH. pH or the extent of alkalinity of their environment is another physical requirement for bacterial growth. Alkalophilus are bacteria that grow in the pH ranging from 8.4 to 9.0 are called alkophilus. Meanwhile, most medically important bacteria are neutrophils, surviving at pH of 6.5 to 7.5. Notice that most human tissues would have a pH of 7 to 7.2. Acetophilus grow in pH less than 6. And these bacteria are called acidophilus. Then the next is the osmotic conditions or the salt concentration. Osmotic conditions will also affect bacterial growth. Normal microbial cytoplasmic salt concentration is about 1%, and for a bacteria to grow, the external environment should have roughly the same salt concentration. If there is higher salt concentration in the environment, water will tend to go out of bacterial cell and the cell will shrink. But if the external concentration um, of salt is low, water will come inside the microbial cell and the cell will swell and may ever rupture. Organisms that require high salt concentrations are called halophilus. Those that require high osmotic pressure for growth are called osmophilus. Then the next is the bacterial growth curve. When you talk about bacterial reproduction, this is actually a forward process which each cell splits into two identical new cells. This process is called binary fission. Individual bacterial cells can divide and the bacterial population can double very quickly in as little as 20 minutes in some species. Then, however, bacteria do not continue growing at such a rapid rate. Indefinitely, this is because factors such as the availability of nutrients and rising toxin levels start to have an effect on bacterial growth. Bacterial growth, there are different stages or phases of bacterial growth. There are different stages of phases of bacterial growth, which is the lag phase, the log phase, stationary phase, the death or decline phase. When we say lag phase, during the lag phase, bacteria are adapting to the environment nutrients and are plentiful and the cells grow in size without dividing, the cell number remain constant. Then the log phase, the exponential phase or the log phase marks a big increase in cell number. Maximum growth rate is achieved with constant doubling of the bacterial of population. Growth then slows as nutrients become depleted and material-based products build up to toxic concentrations. Then the stationary phase is the bacteria that enters the stationary phase when stationary phase. The bacteria then enters the stationary phase when the number of new cells equals the number of cells dying. The total number of cells in the population remains constant. It is during the stationary phase that some bacterial cells capable of forming endospores undergo sporulation death phase. This is the period of rapid cell death where the number of dead cells is greater than the number of living cells. That death phase, more cells die than are produced and the number of cells in the population declines. The duration of this phase varies from a few hours to a few days, and any species that were able to form spores or endospores will survive at this point.